usually a piece of DNA that carries a sequence of DNA or other genetic material and introduces into a new cell. Vectors act as a vehicle to transfer genetic material from one cell to the other cell for different purposes like multiplying, expression, or isolation. Vectors are used as a tool in molecular cloning procedures so as to introduce the desired DNA insert into a host cell. The DNA insert that is transmitted by a vector is the recombinant DNA, and the process is also known as a recombinant DNA technology. Usually, the vectors are DNA sequences that carry different parts which involves in different functions. Vectors usually have an insert, also known as transgene, that carries the recombinant DNA, and a large sequence called the backbone of the vectors responsible for the structure of the vector. So that was the vector and features or characteristic of vectors. Vectors should be capable of replicating autonomously, which in turn depends on the presence of particle sequences in the vector that enables them to initiate replication and propagation within the host cell. Some vectors might even have sequences that allow the production of protein essential for inserted DNA, regulation of the process, and further transfer of insert between the different vectors. The size of an ideal vector should also be small enough for it to be incorporated into host gene. The small size of the vector also enables it to incorporate a large size insert of for transfer. Vectors should be easily to isolate and purify as these need to be recovered and reused for multiple processes. For a vectors to be effective, these should also have certain components that facilitate the process of determining whether the host cell has received the vector. Most vectors should used in the process have a gene that either provide resistant to an antibiotic or produce a particular type of protein. So these components are called marker genes. Many vectors also require unique restriction enzyme epigenetic site that enables the insertion of the vector DNA in the presence of specific restriction enzyme. However, many vectors have been designed with a series of restriction sites close to multiplying cloning site that includes the possible restriction enzyme that can be used to digest the sequence. So the first type of vectors are the cloning vectors. So there are many varieties of cloning vectors like plasmid vectors, bacteriophage, phage. So we are just concentrating on just three. The first three was which what I got, which is bacteriophage, plasmid, and cosmids. So, Cloning vectors are vectors that are can that are capable of replicating autonomously and thus are used for the replication of the recombinant DNA within the host cell. Cloning vectors are responsible for determination of of which host cells are appropriated for replicating a particular DNA cell. Cloning vectors are a further different type that are defined by different features unique to each type of vector. So first cloning vector is plasmid. Plasmids are like extracondylar circular DNA, a molecule capable of replicating autonomously within the host. So it is a vector which can be insert our gene of interest and produce our recombinant DNA, which is RDN. So these are also termed as workhorse cloning vector in recombinant DNA technology. Plasmids can carry insert DNA that is less than 20 kb as a cloning efficiency, and it's according to the gene of interest size actually. So the autonomous replication of the plasmid is made possible by the presence of genes and sequence that can initiate plasmid replication independent of the host replication site. So two type of plasmids are like example are PBR322 plasmid and PSC101 plasmid. So the next one is cosmid. Cosmids are hybrid vectors composed of plasmid and phage lambda vectors capable of incorporating up to 42 kb of DNA. So cosmid is a modified version of plasmid. So the plasmid which contain a cos site is also known as cosmid. So cosmid vectors are created in order to incorporate large size DNA molecule that cannot be carried by plasmid. So these can carry DNA sequence having size ranging from 28 to 46 kb. Cosmid vectors do not have many phase characteristics except the signal sequence that promote phage head stuffing. The hybrid structure of plasmid enables the phage head to incorporate within all the DNA for transfer. So a main classic example for plasmid vector is C2RP. So the next one is bacteriophage vector. So bacteriophage vectors are viruses that only infect bacteria and transform them efficiently while carrying large inserts. Insert means genome. So, the use of phages also facilitate the isolation of large quantity of DNA that can be used for analysis of inserts. Even though there are a number of phages that can and have been used as a vector, phage lambda is the most convenient cloning vector. The use of bacteriophage vector that can incorporate large DNA segment decreases the number of clones required to obtain a particular DNA library within the entire genome of organism. So, some examples of vectors are lambda phase, N13 phase, T4, T7 phase, P1 phase. And so, this is the structure of each plasmid, I mean vectors. So, plasmid here, this is the gene of interest, MCS region, ORI region, uh, selectable marker means marker site. In plasmid, ampicillin or tetracycline will be the most seen marker site. So this is the plasmid. So just like as a plasmid, which contain a cosite over here. So it also contain a marker region, which is ampicillin. So this is the structure of bacteria. So thank you for seeing the video. Like, share, and subscribe.